You are not imagining it? The brain fog. The slow thinking. The moments when a word you have known your whole life simply will not come. The memory that used to be sharp that now feels like it is working through fog. The low mood that arrived gradually and never quite left. These are not signs of aging. They are not signs of weakness. They are signs of something happening inside your brain at the cellular level. Something that has a name, a mechanism, and a food-based solution that most people never discover. Chronic inflammation inside your brain. Scientists call it neuroinflammation. Research published in Current Nutrition Reports in April 2026 confirmed that cellular mechanisms related to the central nervous system, particularly glial cell dysfunction associated with astrocytes and microglia, are among the primary drivers of the cognitive impairment that millions of people are experiencing and being told is normal. It is not normal, and it is not permanent. Welcome to Cellfresco. One belief drives everything we do here. Every cell has a healing food. We do not make food lists. We explain what food says to your cells. Because every meal you eat is sending biological instructions to the cells in your brain. Some instructions promote neuroinflammation, others promote neural repair. Today we go inside your brain to show exactly how neuroinflammation may be causing your brain fog, memory difficulties, and mood disruption, and the specific foods that studies indicate may deliver healing signals to the cellular mechanisms driving it. Here is exactly what we will cover today. First, the research. What 2026 neuroscience confirms about neuroinflammation and cognitive impairment at the cellular level. Second, 3 specific cellular problems that chronic brain inflammation may be creating. Third, 3 specific foods and how studies indicate each one may target a specific cellular mechanism. Fourth, what to eliminate and exactly why each one appears to worsen neuroinflammation. And finally, your daily protocol and action plan. Now the story. Your brain contains approximately 86 billion neurons. But neurons are not the only cells in your brain. They are outnumbered many times over by cells called glial cells. And among the most important glial cells for your cognitive health are microglia. Microglia are your brain's immune cells. In a healthy brain, they are constantly patrolling, monitoring for damage, infection, or dysfunction. When they find a threat, they activate. They release inflammatory molecules, including cytokines and reactive oxygen species. They clear the threat, and then they deactivate, returning to their surveillance state. This is exactly what is supposed to happen. It is one of the most elegant protective mechanisms in the human body. But research from multiple neuroscience institutions confirms that in the modern human brain, chronically exposed to processed food, blood sugar dysregulation, sleep deprivation, and psychological stress, microglia become chronically activated. They do not deactivate. They remain in a perpetual state of low-level inflammation, releasing a continuous stream of pro-inflammatory cytokines, disrupting synaptic transmission, impairing the formation and retrieval of memories, and affecting the neurotransmitter systems that regulate mood and motivation. Research published in 2026 by Better Life Lab identified chronic neuroinflammation as the #1 driver of brain fog, directly slowing neural communication and memory formation. And research published in the journal Cephalalgia found elevated interleukin-6 levels in the cerebrospinal fluid of patients with chronic cognitive impairment, confirming that the neuroinflammatory process is measurable and real. 3 cellular mechanisms, each one driving a specific aspect of what you experience as brain fog, memory difficulty, and low mood. The goal is not simply to think better. The goal is to change the signals your brain cells are receiving every day. Cellular problem 1. Chronically activated microglia may be disrupting synaptic transmission and memory formation. When microglia are chronically activated, they release TNF-alpha, IL-1 beta, and IL-6 into the brain environment surrounding neurons. Research confirms that these inflammatory cytokines can directly impair the process of long-term potentiation, the cellular mechanism by which memories are formed and consolidated in the hippocampus. When long-term potentiation is impaired, the brain struggles to encode new memories and to retrieve existing ones. The cognitive experience is exactly what millions of people describe: the word that will not come, the name you cannot place, the conversation you cannot quite recall. Chronically activated microglia may also prune synaptic connections more aggressively than in a healthy brain. Synaptic pruning is a normal process in development, but research indicates that inflammation-driven excessive pruning in adults may contribute to the progressive cognitive difficulties associated with neuroinflammation. Cellular problem 2. Neuroinflammation may be disrupting the neurotransmitter systems regulating mood. Research confirms that chronic neuroinflammation may significantly affect serotonin, dopamine, and noradrenaline systems. Inflammatory cytokines produced by chronically activated microglia can activate an enzyme called IDO, indoleamine 2,3-dioxygenase, which diverts tryptophan away from serotonin production toward the production of compounds called kynurenines, some of which are themselves neurotoxic. The result may be reduced serotonin availability in the brain, reduced dopamine signaling, and the characteristic low mood, reduced motivation, and emotional flatness that often accompanies chronic neuroinflammation and is frequently misdiagnosed as primary depression. Research published in 2026 confirms that patients with elevated neuroinflammatory markers show measurably different neurotransmitter profiles compared to patients without neuroinflammation. The mood disruption may feel psychological. But research increasingly suggests that part of the problem may be cellular. Cellular problem 3. Neuroinflammation may be impairing the brain's energy supply. Your brain cells run on a fuel called ATP. Research published in 2026 by Better Life Lab confirmed that when neuroinflammation is chronic, the mitochondria inside your brain cells cannot produce enough ATP, and you experience this as mental fatigue, slow thinking, and difficulty concentrating. Research confirms that inflammatory cytokines can directly impair mitochondrial function in neurons, reducing their capacity to produce the energy they need for the intensive cognitive work of memory formation, focus, and problem solving. A brain running low on cellular energy is a brain running on cognitive reserve, and that reserve runs out faster than you might expect. 3 cellular problems. 3 specific foods that studies indicate may help. Food 1. Blueberries, the microglial calming signal. Blueberries are among the most researched foods in relation to neuroinflammation and cognitive function. They contain anthocyanins, and these compounds appear to cross the blood-brain barrier, one of the few dietary compounds that can do so effectively. Once inside the brain, anthocyanins have been shown to reduce microglial activation, specifically by inhibiting NF-κB signaling in microglia. They appear to reduce the production of TNF-alpha, IL-1 and IL-6 from activated microglia. And research from multiple studies suggests that regular blueberry consumption may be associated with improvements in memory performance, processing speed, and the markers of cognitive aging in human trials. Research published in the Journal of Agricultural and Food Chemistry confirmed that blueberry anthocyanins can reach the brain and accumulate in regions associated with learning and memory, including the hippocampus and cerebral cortex. Every cup of blueberries may be delivering anthocyanins directly to your microglial cells, where studies indicate they can help reduce the chronic activation that is impairing your synaptic transmission and memory formation. 1 cup of blueberries every single day. Your microglial calming protocol. Food 2. Fatty fish, the neurotransmitter restoration signal. The single most powerful dietary intervention for neuroinflammation and neurotransmitter function is omega-3 fatty acids from fatty fish. Specifically DHA, docosahexaenoic acid, the primary structural fat of the human brain. DHA from salmon and sardines incorporates directly into neuronal cell membranes, maintaining their fluidity and supporting the efficiency of neurotransmitter receptor function. Research from the 2026 Cognitive FX study confirmed that omega-3 fatty acids from marine sources, particularly EPA and DHA, can support the brain's natural anti-inflammatory processes through specialized pro-resolving mediators including resolvins and protectins. These resolvins and protectins appear to actively signal microglia to return from their activated inflammatory state to their surveillance state. They do not merely reduce inflammation, they actively drive its resolution. And research indicates that DHA may also support serotonin receptor sensitivity, potentially supporting the neurotransmitter pathways that chronic neuroinflammation has been disrupting. Every serving of fatty fish may be delivering DHA directly to your neuronal membranes while activating the pro-resolving pathways that signal your microglia to stand down. Salmon or sardines 3 times per week. Your neurotransmitter restoration and neural anti-inflammatory protocol. Food 3. Walnuts, the brain energy restoration signal. Your brain cells need energy to function, and chronic neuroinflammation is impairing the mitochondria that produce that energy. Walnuts are among the most studied whole foods for supporting healthy brain function and protecting neurons from oxidative stress. Walnuts contain ALA, alpha-linolenic acid, and omega-3 that research suggests may be converted in the brain to DHA, providing additional neural membrane support. But more specifically, walnuts contain vitamin E in its most bioavailable form, polyphenolic compounds including ellagic acid and folate. And research suggests this combination may support mitochondrial function in neurons by reducing the oxidative stress that inflammatory cytokines generate inside brain cell mitochondria. Research published in multiple studies suggests that regular walnut consumption may be associated with improved cognitive performance across multiple domains, including memory, processing speed, and executive function. And research from the 2026 Brain Fog Science Review identified nutrient support for mitochondrial function as one of the most evidence-backed approaches to improving neuroinflammation-driven cognitive impairment. Every handful of walnuts may be delivering the nutrients that appear to support the energy-producing machinery inside your brain cells, supporting the cognitive function that neuroinflammation has been impairing. A small handful of walnuts every single day. Your brain energy restoration protocol. Now your daily protocol: 3 foods, 3 cellular signals. Every day, morning, 1 cup of blueberries with breakfast. Your microglial calming signal. Anthocyanins crossing the blood-brain barrier and reducing the microglial activation, impairing your memory and synaptic transmission. Midday, a small handful of walnuts as a snack. Your brain energy restoration signal. Supporting the mitochondrial function inside your neurons that neuroinflammation has been impairing. 3 times per week, salmon or sardines for dinner. Your neurotransmitter restoration and pro-resolving signal. DHA incorporating into your neuronal membranes. Resolvins signaling your microglia to stand down. What to eliminate and why each one appears to worsen neuroinflammation. Sugar and refined carbohydrates produce blood sugar spikes that research confirms activate microglial inflammation through AGE receptor signaling. Ultra-processed foods are rich in omega-6 fatty acids that research indicates directly fuel the arachidonic acid pathway, producing the most damaging neuroinflammatory molecules. Alcohol in excess has been shown to activate microglia and increase neuroinflammatory cytokine production, worsening exactly the synaptic and neurotransmitter disruption described above. Poor sleep is now confirmed as one of the most powerful drivers of neuroinflammation. Research confirms that just 1 night of poor sleep measurably increases brain inflammatory markers the following day. Chronic psychological stress activates microglial inflammation through the same HPA cortisol pathway we discussed in the context of belly fat. These are not lifestyle suggestions. They are direct inputs into the neuroinflammatory cycle that may be impairing your thinking every single day. Your action plan: 3 cellular problems, 3 healing signals, 3 opportunities to help your brain cells move back toward balance. Blueberries every day. Your microglial calming and NF-κB suppression signal. Walnuts every day. Your brain mitochondrial energy support signal. Fatty fish 3 times per week. Your DHA neural membrane and pro-resolving microglial signal. Start tomorrow morning. Blueberries with breakfast. Walnuts as your midday snack. Salmon for dinner this week. Your brain fog may not be aging. Your memory difficulties may not be inevitable. Your low mood may not simply be who you are now. They may be the cellular expression of a brain struggling with chronic inflammation. If this video explained what your doctor never connected for you, please give it a thumbs up. It helps more people find this channel, and it means everything to us. Subscribe to Cellfresco. Every cell has a healing food, and every week we show you exactly which one. Keep healing, keep thriving,